Hello, everybody, and welcome to Creation Myths. As far as I can tell, we are live. So what I'm going to do is just take a second here, make sure everything looks good on the YouTube side of things, and then we can get started. Very happy to have everybody here. All right, let's see. This appears to be working. Delightful. Okay, so if at any point, if there's a problem where the slides are not moving, if things look glitchy, if you can't hear me or anything, just let me know in the chat and I will fix that as soon as possible. This is, as always with me, this is a little bit seat of my pants, so we're just going to go with it. So thank you all for being here. Tonight, what we are doing is we are going to review this book, Universal or Separate Ancestry, the biblical model of origins made easy, the question of ancestry finally answered. That's the full title by our very own YouTube Young Earth Creationist, Standing for Truth. Now, a couple of little logistic things before we get into it. I did invite Standing to be here so that I could go through it, and then if you wanted to hop on and talk about it, we could do that. He is not available tonight, so we could not make that work. But perhaps maybe down the road we can have a conversation about this if he wants to. Second thing is that you may have previously seen a book club kind of roundtable discussion on this book that our friend Dapper Dino hosted over on his channel. I participated in that. That was more just our kind of big picture thoughts on the book. We didn't have time to get really into the nitty gritty of the science. I mentioned some of the problems with the science in it, but it wasn't really the appropriate venue to like really get into the nuts and bolts of why the scientific claims are wrong. So what we're doing here is I'm not so much reviewing kind of the book as a unit. What I want to do here is go through some of the major scientific claims in the book and see how they hold up. And as you might expect, if you watch that conversation, or if you've watched any of my videos or seen me talk to Standing for Truth, as you might expect, the scientific claims in this book hold up not even a little bit. And I want to make that clear in some detail tonight, because we're going to go through and look at just some of the major claims in here. Now, we're not going to cover everything. There's stuff in this book that I'm not well versed in. So like there's geology stuff. I'm not a geologist. I don't know about the geology stuff. Fossil stuff. Also not well versed in the fossil stuff. But fortunately, most of the arguments in this book are genetic in nature, and that is stuff that I am qualified to comment on. So what we are going to do is kind of go through some of those major arguments. Now, that being said, I'm still going to omit things, and I'm going to kind of give short shrift to a few things just because there isn't time and I don't want this to take forever. I figure I haven't timed this out. I'm figuring maybe like by 9 if it goes really long, but I don't think it's going to be all that long. So what I'm going to do here is, rather than go through this book kind of in order, what I'm going to do is go through it in terms of the topics. And that's one of the problems with this book, is that it jumps around thematically. There are chapters, but there is not a lot of rhyme or reason in terms of what is in each chapter. So I'm going to be showing like pages from the books that are related to specific topics, and you'll see it jumps around. It'll be in the 20s, then the next mention of that topic will be in the 50s, and that's just kind of how this book is structured. So don't worry if you have the book. Don't try to follow me like kind of as we go through. I'm going to be talking topic by topic, and we'll go from there. So the last thing I want to say before I get into it is that if we have time, I don't want this to go too long, but we'll see how long it takes to kind of go through what I have set up here. And then if we have questions or anyone wants to contribute anything else at the end, we can do that. But we'll see how much time we have left. We'll see how we're looking by 9, and we'll go from there. So that being said, let's jump right into this. The first thing we're going to talk about is human genetics and human origins. A large part of, no, Dapper, you're not late. Well, I guess you are late. I started on time, like you. And I'm trying to do that. So that's what we're doing here. So you haven't missed any of the content though. So you just missed the intro spiel. We're now just doing content. So we're into it. So we'll start with human genetics and human origins. A very large part of this book is dedicated to trying to argue against the standard model of human origins, which is to say the out of Africa model descended from other hominins in the neighborhood, modern humans appearing in the neighborhood of 200 to 300,000 years ago. So what I'm going to do is I'm going to show you some pages and some claims, and then we're going to go through and we're going to evaluate that stuff just to kind of let's really dig into the science that is presented in this work. Okay, so first here, right on page 7, if you look on the left side right here, we can see what would you honestly expect to find if Adam and Eve were true? After examining this question, we should expect evidence of one female ancestor of all people on the planet, as well as evidence for one male ancestor of all people on the planet. Genetic data confirms these expectations. That's claim number 1 there. Genetic data confirms these expectations. Now, is that what we should expect to see? Is that what the genetic data confirm? No. This is how most recent common ancestors work. What Sting is alluding to here is the concept of mitochondrial Eve and Y chromosome. It's actually Y chromosome Noah, although everyone calls him Y chromosome Adam, but it would be Y chromosome Noah, which is the most recent common ancestor for the maternally inherited mitochondrial genome and the paternally inherited Y chromosome. Now, the thing with most recent common ancestors, a very common misconception, is that they're the only individual alive at the time. But that's not the case. If you look at this figure, and the reference for this figure is in the bottom here, and I'll also put it in the description. I just haven't updated the description with all the links I added yet. If you look at this figure, this blue dot right here represents the most recent common ancestor of this living population at the bottom. So if each of these dots represents an individual, at the top we have the past, at the bottom we have the present. This blue dot represents the most recent common ancestor for this living population. You could trace all these individuals back, and they all go right here. Actually, the most recent common ancestor is this green dot right there. That's the most recent common ancestor, sorry. Now, if you go to a different generation, you get a different most recent common ancestor. So if you go to this generation, or this generation, or this generation, your most recent common ancestor ends up further in the past. The other thing is that you'll notice the most recent common ancestor is not the only individual that's alive at the time. There's lots of other individuals alive at the time. And in fact, those other individuals also contribute genetic material to the extant, the living generation. However, when you're tracing one specific piece of the genome, you might miss the part that those other living individuals contributed. And we know this is the case. So for example, the X chromosome, most recent common ancestor, existed like 500,000 years ago. And you could do this kind of thing for every part of the genome. You could trace backwards and find a most recent common ancestor. The only ones that converge at the times for mitochondrial Eve and Y chromosome Noah are the mitochondrial DNA and the Y chromosome. The rest of the genome converges further in the past. And the reason for that is the rest of the genome is more diverse. It's diploid, right? Rather than being inherited from a single parent, you inherit it from both parents. So it just, given a population size, you have more variation and it takes longer to converge back in the past to those points. So we're not going to go through the math right now at this moment, why the calculations for most recent common ancestor are wrong that creationists use, putting them at about 6,000 years ago. We'll do that in a minute. But even putting that aside, it's just the mitochondria and the Y chromosome. The rest of the genome, even if you use the creationist math, converges further in the past. And there weren't just two individuals alive at the time of those most recent common ancestors. Now, I do need to say here, this is a theme throughout this book, so I'm going to say it here, but it applies elsewhere. What's going on here is that Standing is not making his own argument. He's taking a well-trod creationist argument. He's getting an argument from the professionals, and it's kind of going through a filter of a layperson who is, not a biologist, and that's fine. There's nothing wrong with that. But it adds to the possibility of misunderstanding and confusion in the wording. Now, in fairness to what Standing is dealing with here is he is not the person making this misconception. His sources are making this misconception. So this is a direct quote from a 2018 paper by Carter, Lee, and Sanford. I don't know who the Lee guy is, but that's Rob Carter, Dr. Rob Carter, Dr. John Sanford, prominent young Earth creationist biologists. And they said, for years, geneticists have known that there is a single paternal ancestor and a single maternal ancestor for all of humanity. That sentence alone is something you read in papers. That's not a problem. The problem is the next sentence. This is a direct prediction of the biblical model, meaning they're referring to one specific male and one specific female. That's not what a most recent common ancestor is. And, you know, there's an open question of the degree to which one could like fault somebody like Standing for Truth, or, you know, any one of the non-expert creationists that participate in this discussion. But you cannot give that same benefit of the doubt. Carter and Sanford, and I don't know who this Lee is, but I'm assuming he's also professional. They know what a most recent common ancestor is, and they're just lying to their audience right there. So that's the first problem that's going on here, is that yeah, the information in the book is wrong, but it's wrong because the sources are lying to the young Earth audience. The sources, the professionals, they know better. So now we have on the right here, don't worry about reading that. This is how the references are done in this book. It's like screenshots of like the headlines and maybe a paragraph, or it's screenshots of a reference, or in at least one case, screenshot of Wikipedia. So this is how the references are done. But basically, this is a paper that purports to show, this is a paper from 1998 that purports to show that the human mitochondrial most recent common ancestor existed 6,000 years ago based on the mutation rate that they're calculating in this specific paper. Now, if you jump forward a bit to chapter 5, we can look at this first paragraph right here. The reason for this is because pedigree-based studies and empirical methods reject deep time evolution and universal common ancestry. The best tool to determine what is related and what is not is molecular clocks and DNA function. Now, what they are saying here is you directly observe the rate of mutations in like a pedigree study. Like you look at a mom and you look at a daughter and you compare the differences. That's a pedigree study. You can count the mutations that are different between them, and they're saying, well, that's the empirical rate. And what they're saying, if you jump up to this paragraph at the top right over here, is that evolutionists look to what is called time dependency to explain away the data. They have suggested that the mitochondrial DNA clock ticks at different rates at different points in history. Basically, the claim that Standing is making here is that biologists, when we calculate the time to most recent common ancestor, how long it would take for a given observed level of diversity to appear in extant living things, how long do you calculate backwards to a common ancestor? He's saying we invoke this thing called time dependency, where the mutation rate actually slows down. Okay. Now, again, he's not coming up with this. That's from Nathaniel Jeanson in his book, Replacing Darwin. This is specifically the thing that Jeanson says, that those stupid evolutionists think that the mitochondrial mutation rate slows down as you go backwards in time, and evolutionists, dumb evolutionists, or those dishonest evolutionists are invoking this unobserved rescue device to make it so that the most recent common ancestor is 200,000 years ago instead of 6,000 years ago. Now, the problem for creationists here is that the mutation rate does not change over time. The thing that changes is the number of mutations that persist over generations within a population. So let's look at the different ways we can measure changes. And by the way, I apologize to anybody who wasn't expecting what is accidentally turning into a biology lecture here, but I'm finding it impossible to not fall into professor mode. So that's where we're going with this, apparently, and I apologize, but that's what we're doing. So the fastest rate you're going to look at, if you're looking at one individual, the fastest rate you're going to look at is called mutation rate. It's mutations per site per replication. So every time the genome duplicates, how many changes per site occur? That's a mutation rate. And I apologize, I have a cold, so I have to wipe my nose. I'm going to do it off camera, but I'll be back in just a second. All right, my apologies. So that's the fastest rate. That's just talking in one individual as your DNA replicates. How many errors occur? How fast do changes happen? Okay, but when you do a pedigree study, you have reduction in those mutations. You have the germline bottleneck. So like I have a bunch of mutations in my muscles, in my brain, in my skin. Those mutations are never going to make it to my kids because they're not in my germline cells. So you have a germline bottleneck, right? The other thing that happens is within an individual, you can have selection and you can have genetic drift. A cell that experiences a mutation that is fatal will never pass on its suite of mutations to subsequent generations. And sometimes, like in the germline cells, just by chance, some things propagate and some things don't through genetic drift. So you can compare parents and offspring, or parents and parents and offspring, however many generations you want, when you can look directly at the individuals and calculate the pedigree rate. That's the rate at which mutations are inherited from person to person. And that rate is going to be slower than the mutation rate because not all of the mutations that occur within an individual are going to be passed on to their kids. Okay, so that's the pedigree rate. But it slows down even further over long periods of time because you have processes like selection, genetic drift, and inbreeding that are going to further reduce diversity within a population. And over many, many generations, that's going to lead to the phylogenetic rate. This is the rate at which mutations accumulate within a population, the phylogenetic rate. So now we're looking over long periods of time, okay? But we're still not done yet because the way that you really want to look at long-term differences in populations, what you want to look at is the rate at which mutations experience fixation, the rate at which a specific mutation becomes present in 100% of the individuals within a population. That's called the substitution rate. And the units are substitutions per site per year. So for every site in the genome, how many changes are there per year within that population? That's substitutions per site per year. That's the slow rate, that's substitution rate. When you're doing long-term calculations, when you're calculating the time to most recent common ancestor, you use either the phylogenetic rate or the substitution rate, depending on the context. When creationists do this math, they use either the mutation rate or the pedigree rate, which is wrong. It's artificially fast. And we can look at this visually, and this figure comes to us from Evograd on his wonderful series, which I think is really the definitive written takedown of Nathaniel Jeanson's Replacing Darwin. So again, the link is on the slide, and I'll also link to it in the description. But this is a figure of his that illustrates this trend in the decrease in the apparent rate over time due to those filters that I talked about, the germline filter, inbreeding, selection drift, that kind of stuff. So you have Jeanson's rate up here. This is the one that creationists love to use. But then you look at other human pedigree studies, and they're longer than that. So you have slower rates in other pedigree studies. And then if you look to the phylogenetic and substitution rates for human origins, human Neanderthal, and human chimp divergence, you end up kind of asymptotically approaching what is that true long-term substitution rate. Okay, so this is the problem that that we find in the math that's presented by Standing for Truth in here. It's taking stuff from the professional creationists that is just straight up wrong, and this is why it's wrong. It's very simple to understand why it's wrong, and presenting it as though it is set in stone fact. And again, I do have to say, not Standing for Truth's fault. He's not an expert, but Nathaniel Jeanson's an expert, and you better believe that Jeanson, with his fancy Harvard PhD that we are reminded about all the time, he knows the difference between these things, and he knows why what he is doing is wrong. And he actually, in one of his papers, AIG, actually does so. He kind of lets the masks up a little bit. So that's why this section is incorrect here. So now let's turn our attention to the out of Africa hypothesis, because, yeah, I know I'm going to get evolutionary assumptions in response, and the answer to evolutionary assumptions is that, like, there are other data points in here that are based on archaeological evidence and the historical record. And there's a paper that I like that Standing and Rawmat have talked about that I presented as saying, look, here's predictions that have been confirmed. And they say, oh, those are predictions, but they can't read the paper. And I'd be happy if either of you end up watching this to do a journal club with you with that paper, and we can go through it and talk about it. But basically what you can do is you could say, well, we know when the island of Vanuatu was built. We know when the Canary Islands were settled. We know archaeologically when Japan and when Europe were settled. So what we can do is we can, given the divergence times between, say, the Vanuatu population and the sister mainland population, we know the divergence times, we can look at the genetics and see the differences, and we can see where they fall on this curve, given that we know where the time before present is. And we can do that for a number of data points going back tens of thousands of years. So those islands I mentioned were settled in the neighborhood of 2,000 to 3,000 years ago. Europe was settled 45-ish thousand years ago, Japan 32-ish thousand years ago. And what the authors of this paper did, and if I remember, I'll link it in the description, is they said, okay, we know these dates, so let's do the genetic math and let's see if the expected number of differences is what we actually find. And the answer was yes, it's what we actually find. And now some of those creationists can say, oh, well, those are assumptions. Some of those are like within recorded human history and they're not assumptions at all. So that's the response to that. So now let's look at the out of Africa hypothesis, which if you're going to contest human evolution, you're also necessarily going to contest the out of Africa hypothesis. So what we want to do here is look at kind of the big problems, and then we'll look at the kind of specifics and a little bit of the creationist. I'm learning to love the phrase rescue device, so we're going to look at that. Basically, what Standing is saying here, I'm going to spare you all reading all of this, but I'm going to put it on screen. So if you want to read it now or read it later, you can. Basically, what the argument here is that the genetic structure of human populations, where you have serial bottleneck events as you have founder events, as migrations have radiated outwards from like East Central Africa, that is incompatible with the creationist theory of created heterozygosity. Now, this is, again, something you're going to read about in Replacing Darwin by Nathaniel Jeanson. And by the way, I will have one of these on that book at some point, maybe have to do more than one. And the problem initially is that you're assuming created heterozygosity without any good reason for doing so. Now, he also says that you have low genetic diversity within humans, which is consistent with a recent creation. As we'll see, there's a bit of a contradiction there because later on he says that we're pretty diverse. So it's a little bit weird there. But if you look at page 70, oh, here we go. If you look at page 72 here, this is where there's like an attempt to actually explain the population genetics of what's going on here. The big idea for the creationist model is that you're starting with the assumption of a Middle East origin and then layering on things from there to make it work. So layering on things like created heterozygosity and weirdness with the population structure. So let's look at why these arguments are wrong. Actually, let me just read a little bit right here. Up here in the top right, we have multiple lines of evidence indicating this was the case. God created Adam, Eve, and the kinds with lots of genetic potential. So that's created heterozygosity. That's why you don't have like very little diversity from various bottlenecks. And then we have remarkable created and useful heterozygosity, even a tiny percentage of the nucleotides through the process of gene conversion and recombination, an effectively unrestricted range of beneficial diversity would be generated. Okay? So that's kind of the idea here. So let's just look at how we know that humanity originated in Africa. Putting aside the timeline, let's just look at how we know we originated in Africa and how it conflicts with the creation model here. So lots of lines of evidence. There is diversity and there's phylogeny. So when we say diversity, what we mean is that there's more genetic diversity among African populations than there are among any other populations anywhere else in the world. The two most genetically divergent people in the world both live in Africa, or I should say both have recent African ancestry, okay? And the reason for that is anytime you leave, you have a population, anytime you have a founder event or a migration away from that population, you're taking a subset of diversity within that population and taking it with you into the new area. So you migrate from Africa into the Middle East into Central Asia into Europe and East Asia, and so on and so on. Every one of those steps results in a decrease in genetic diversity in the subsequent population. So you have the most diversity in Africa, and then it lessens as you leave. Phylogenetically, we can look at this and we can see that African lineages form the root of the Homo sapiens tree. And you can see that in this figure right here. The red represents African lineages. So you can see that the root, about 200,000 years ago, representing the origins of humanity, that is going to be African populations. And then descended from that are all of the various non-African lineages in subsequent migrations out of Africa. Okay? You can't say that we originated in the Middle East. You can have all kinds of funky mechanisms to try to make that work in terms of the genetic diversity, but it doesn't solve the logic problem that all the outside of Africa diversity nests within the African diversity. Third problem, heterozygosity. Heterozygosity is something that is going to decrease with those migrations and founder events, because again, heterozygosity is a proxy for genetic diversity. So as you leave Africa and you migrate outwards, you're losing heterozygosity. You're increasing the homozygosity of your population. This figure shows very neatly what's going on here. The y-axis is haplotype heterozygosity, so higher is more diverse. And the x-axis is really clever. It's distance to AA, and AA means Addis Ababa, which is the capital of Ethiopia. So what this shows, and then it's color-coded, right? So red is Africa, you've got Middle East, Europe, East Asia, and then Oceania and the Americas. And what you can see is that as you follow those migratory trails out of Africa, you get less and less diverse. The really cool thing is this is a nice linear relationship that points right back to East Central Africa, which is where humanity originated. This evidence is cut and dry. We came from East Central Africa. You can't shoehorn a Middle Eastern origin into these data. And then the fourth thing that I'll mention is called linkage disequilibrium. Now, this is a little bit complicated, so I'm going to do this by way of analogy. Think about a deck of cards, right? You have a deck of cards that is new. Okay, it's completely unshuffled. You've got all the cards all in a row by suit and then by number, right? So initially, it is at disequilibrium, right? The 3 of hearts is very likely to be near the 4 and the 5 of hearts initially. Those things are likely to be linked together within that deck of cards. But as you shuffle that deck of cards 10 times, 100 times, 1,000 times, 1,000,000 times, you lose that disequilibrium, and any card is equally likely to be next to any other card. We can look at the same thing in our genomes. And what you find is that genomes that are at linkage equilibrium, meaning different parts of those genomes are equally likely to be associated with other parts in the same genome, in the same individual, right? That is the hallmark of a population with a lot of diversity that has been in existence without any loss of diversity for a prolonged period of time. We find the highest levels of linkage equilibrium within African populations and the lowest levels of equilibrium and the highest disequilibrium outside of Africa, indicating that those populations outside of Africa have been affected by things like genetic drift and inbreeding that are going to decrease diversity and maintain linkage blocks throughout time. Okay, so that's another line of evidence, a fourth line of evidence that points to an African origin for humanity. Now, of course, creationists are not silent on all of this, and you'll read about some of the things in this book that supposedly address these problems. So first, there's just no evidence for created heterozygosity. There's just no evidence for that. It's a made-up thing that we don't see. And also, it doesn't explain the degree of diversity that we find. For some human genes, there's just a lot of alleles, and you can't explain that through an original created pair, Adam and Eve, and then go from there. There's just not enough diversity to go around. The other thing that you see from creationists is that you're going to invoke faster mutation rates among African lineages. So this is another thing that is a big problem. The slope just represents the change in heterozygosity. Sorry, I'll just go back for a second. The slope represents just the change in heterozygosity over time, over distance from East Central Africa. The intercepts don't matter. Don't worry about the intercepts. It's just the maximum heterozygosity we find, and then the distance from Addis Ababa is basically how far can you get in the world. Yeah, the intercepts don't matter for that one. Okay. So this faster mutation rate among African lineages, this is just made up again. Like, we don't observe this. All humans use the same DNA polymerase that copies our DNA, and you get the same like it's the same. So, you pick 2 random people and they're going to have the same per base mutation rate. But let's assume that you do have faster mutation rates in African lineages. That doesn't solve the phylogenetic problem. The African lineages are still the common ancestor, and then everything is nested within that. What creationists need is for the Middle Eastern lineages to represent the ancestor and for the African lineages to be nested inside that. You could increase the mutation rate. You don't solve that problem. And by the way, this is a confusion that Standing has had before, where we've talked, for example, about Neanderthals, and he's had confusion about divergence versus diversity. And faster mutation rates gets you diversity. But they don't get you divergent points or divergent timers. They're going to be nested within the more ancestral groups. Okay, so that's that problem. Second thing is faster mutation rates don't solve the linkage disequilibrium problem. They do nothing to solve that for you, because that's something that's going on outside of Africa that you need to account for, not within Africa. So there you go. So the takeaway from this section is that if you want to overturn the out-of-Africa hypothesis, you need a better comprehensive model. What creationists do is they look at the diversity and they say, oh, we can solve that by having different mutation rates in different lineages. Oh, we look at the heterozygosity, or we'll look at the heterozygosity versus the homozygosity and say, oh, well, these lineages were inbred, but those weren't. Therefore, that's why we see the difference. It's not about origins, it's about population structure. They introduce all sorts of silly things like that, so you end up layering these ad hoc explanations on top of each other without building a comprehensive model that explains the whole thing. And I should say those ad hoc explanations are not observed. They're actually contradicted by the available data. So that's why the arguments presented here are wrong. The out-of-Africa model, on the other hand, is comprehensive. It is in line with all these things. And I should note, creationists love to point out how evolutionists don't make predictions. Well, the out-of-Africa hypothesis originated via archaeological evidence. And those predictions about the genetics were all made later on and then checked. And those predictions were all confirmed. Things like the phylogenetic structure, the diversity, the heterozygosity, and the linkage disequilibrium. Those were all verified predictions from the archaeological evidence that first pointed to an out-of-Africa model. So just be aware of that, creationists. There's some predictions for you. How's everybody doing? Everybody hanging in there? This is a lot of genetics and such. Everyone good? All 16 of you? Thank you all for being here. So the next thing is an argument that Standing leans on relatively heavily. It's called genetic entropy. And some of you may have heard me talk about genetic entropy before, because I've talked about this a lot. I'm not going to cover this in detail, because I've had 2 debates with Sal Cordova about genetic entropy. Sal Cordova works with John Sanford, who came up with genetic entropy. I have a couple of videos on genetic entropy. I will link all of that stuff below. I haven't done it yet because I'm doing this on short notice here. But I've talked about this a lot, so I'm not going to go through it, but I am going to go through the basic problems. And I will note that Standing for Truth makes no effort to address any of the problems. Basically, he is just presenting the barest bones of what Sanford says and taking it as fact without digging into it and actually, 1, demonstrating that that's how the numbers actually work, or 2, defending them against the criticisms. Okay, and then I'm also going to address some minor specific issues here. Okay, so first thing is kind of a small thing that I want to address here. If you look on the left side of this page down here, well, first, I'll comment on the writing right here. Dr. John Sanford coined the term genetic entropy. Genetic entropy is not a good thing. It's not a fun thing. There is nothing enjoyable about genetic deterioration. When I read that, my mind immediately went to Kent Hovind's Patient. If you haven't read that, look that up. But in a book that's supposed to be presenting a scientific argument, the writing is not up to that standard. But what I want to point out first is just this paragraph right down here. By the time you're in your 60s or 70s, you can have roughly 40,000 new mutations per cell. There's no reference for that. I tried to look up that number and I couldn't find it. The best I could find was a study that showed by the 80s, you could have a few thousand mutations per liver cell. Now, there are cells in your body that divide a lot faster than your liver cells. So it's possible, I suppose, that you could have a cell that has 40,000 mutations. But I could not find a reference that was anything even resembling that. I could not do it. So I don't know where it comes from. I don't know if you read it somewhere. I don't know if there's a reference for it in the back of the book, because there are a handful of references in the back, but I did not go through each one. There's no reference on the page. I got nothing. I couldn't find it. Now, direct your attention to the right side right here, because this illustrates another very common confusion that creationists have when they talk about genetic entropy. So right here, this is a paper looking at genetic meltdown in eastern lowland gorillas. Now, genetic meltdown is what happens when a population gets very small and you have an accumulation of recessive deleterious traits. Okay, so you have traits that are harmful, but they're recessive, but because there's a small population, there's inbreeding, because it's a small population, you end up with a lot of homozygotes for those traits, and then you actually express the harmful traits. Another example of this is the hip problems that you see in cheetahs, same basic idea. What can happen here is that you go into what's called an extinction vortex, where you have small populations, you have genetic drift and inbreeding that leads to these deleterious traits, deleterious meaning harmful, not deleting something, leads to these harmful traits being expressed, and now the next generation is even worse off in terms of their reproductive output, so the population gets smaller, and then you have stronger drift, you have more inbreeding, and it's a vicious cycle until the population goes extinct. That's called an extinction vortex. Now, Standing is putting this forward as an example of genetic entropy. The problem with that is that genetic entropy is the opposite thing. Genetic entropy, the real term for that, is error catastrophe. It's when mutations build up faster than they can be selected out, and a population accumulates lots of harmful mutations that break things, and it ultimately goes extinct for that reason. The fitness drops off over time as mutations accumulate. So genetic entropy is extinction, or ultimately extinction, loss of fitness, due to an increase in genetic diversity. A meltdown, an extinction vortex, is extinction due to a loss of genetic diversity. They're opposite things. And Standing for Truth does not know that they are opposite things, because he's not a population geneticist. He hasn't studied population genetics extensively. But if you're going to write a book on that topic, take 5 minutes to check, to maybe just check, to make sure that the thing that you're using as evidence for a specific process is an example of that thing and not the exact opposite thing. Because what we have here is Standing using something as evidence for genetic entropy that is actually the exact opposite of genetic entropy. That's what's going on here. Okay? That's a problem. Now we can continue. We'll just give you a representative example of what he says here. Selection ought to somehow thwart the accumulation of large numbers or huge numbers of minor mutations. If there is, if there's no type of selection that can remove so many deleterious mutations entering the human population, the population of all living organisms, the species will rapidly decline. Higher genomes will eventually degenerate to the point of extinction. Yes, this is fundamentally a population genetics argument. And I will just shamelessly plug my debates with Sal, where I tried to get him to go through that argument with me, and he was not interested in doing so. And to date, nobody's been really interested in doing so, despite my asking in several venues of creationists to walk through the population genetics with me. Nobody's really interested in doing it. I will say, not to make an argument from authority here, but just so people know where I'm coming from. Error catastrophe and mutation accumulation was the subject of my PhD thesis. This is what I wrote about for my thesis. So when I read a genetic entropy argument, none of it makes any sense, because none of it works with the math that I'm very familiar with to make that kind of thing happen. So now, one thing that SFT does here is he ignores new research. He says right in that last page that these genomes are going to go extinct as mutations accumulate and you can't select them out. He ignores research from this year. This is mutational load in the functional fraction of the human genome. I'm not going to quote extensively from this paper, but I'll just kind of big picture what these authors say is we find that the functional fraction is not very likely to be limited substantially by mutational load. In other words, you can have a highly functional genome, and the number of mutations that occur is not going to mess with it. And that any such limit, if it exists, depends strongly on the selection coefficients of new deleterious mutations. In other words, if the mutations are harmful, they'll be selected out. If they're tolerable, it's fine. They'll kick around. And there's like studies to back this up, like Springman et al. 2010, where they showed that bacteriophages could be saturated with mutations, and the population actually, the maximum fitness actually increases. So then we have another problem here. Sorry, everybody, I'm just going to take a quick break to cough because, again, I still have this cold. Just a sec. My apologies. Thank you all for bearing with me. So what I want to point out here is this figure on the left. This is John Sanford's mutation effect distribution from his book, Genetic Entropy and the Mystery of the Genome. This is a fundamentally dishonest presentation that Standing is just lifting whole hog and just using it as though it is perfectly reasonable. What Sanford says here is that the person who made this distribution, he didn't draw it like this, but he did the math, Moto Kimura, what he said is that beneficial mutations basically never happen. This is what Sanford wrote. Kimura obviously considered beneficial mutations so rare as to be outside of consideration, and that's from page 22 and 23 of the third edition of Genetic Entropy and the Mystery of the Genome. Thank you. Yeah, this is a daycare cold. So now, this is what Kimura actually wrote in the paper that Sanford cites to make that graph. We disregard beneficial mutations. This is an oversimplification, but a model assuming that beneficial mutations also arise at a constant rate independent of environmental changes leads to unrealistic results. Basically, Kimura was modeling neutral evolution, and his model was so simple that mutations can only occur at a constant rate. Beneficial mutations don't occur at a constant rate because you have selection, right? So you get to a fitness peak, and at that point, they stop occurring unless the environment changes. So Kimura was saying, we're going to ignore them. We're not going to build that dynamic into the model. He later said the selective advantage of such mutants, which is to say the neutral ones that accumulate, the selective disadvantage of such mutants will easily be taken care of by adaptive gene substitutions that must occur from time to time. In other words, beneficial mutations, that's adaptive gene substitutions, that means beneficial mutations. So he says right there, beneficial mutations happen. Sanford is just lying about what Kimura said. Like, it's that simple. It's not complicated. Standing also references some rescue mechanisms, but this doesn't make this part of the oh, my slides are a little messed. There we go. They seem to be better now. Standing also references some rescue mechanisms, but he doesn't specify what they are. So it's hard to follow his argument, because if you're not like familiar with the discourse as a reader, then you're going to get lost, because he's referencing things without saying what they are. It's, oh, evolutionists invoke rescue devices to make this work. Okay, what are they, and why don't those mechanisms that you're calling rescue devices work? What I can gather is that we're talking about things like epistasis here, where you have interactions between things. So let's game that out. Let's see, does epistasis actually a problem? He'll claim, and Sanford and others will claim, that epistasis, interactions between genes or mutations, is a problem that actually makes genetic entropy worse. But let's game that out. So basically, if you have mutation 1 and then mutation 2, and they appear sequentially like that, they can interact with each other in terms of fitness, and you have 4 options. 2 of them are what's called synergistic epistasis. So if you go from one beneficial mutation, you get a second one, and it makes everything better, no problem. We got 2 beneficial mutations. Hooray, everyone. If you have one harmful mutation and then another, and in combination they're worse, that's also good, because it's easier to select them out. Basically, it has a larger fitness effect. The other side of this is called antagonistic epistasis. This is where they have opposite effects. If you start with a bad mutation and then another one occurs and the net effect becomes good, okay, great, we've reversed a bad mutation. And then if you, sorry, I'm off again. I really apologize, everybody. Now, the last situation is if you have a good mutation, and then a second one occurs, and it causes it to be bad. But this requires that the beneficial mutation happen first. So if you have a population with the beneficial mutation, then some individuals get the bad one that makes it bad. Well, you haven't removed the beneficial mutation from the population. Uh-oh. I may vanish for a second, but I think I'm okay. And don't worry, we're almost done here. After genetic entropy, it's shorter. So let me just get to the big picture on entropy here. There are 4 questions that are important for genetic entropy. Are fitness effects of mutations constant? If a mutation occurs, does it always have the same fitness effect? The answer is no. You have epistasis, you've got things like the heterozygote advantage, that's like the sickle cell allele. You have environmental differences, it's good over here, but bad over there, right? So mutations can be good and bad. Oh yeah, you're right. I didn't talk about neutrality in the context of epistasis. You're absolutely right. So there's more combinations. Second question for genetic entropy, are there basically 0 selectable beneficial mutations? The answer is, of course, no. You can see this in viruses. You can see this in people, lactase persistence. That's a selectable beneficial mutation. Third question, does the frequency of harmful mutations remain constant even as they accumulate? Absolutely not, because that's just math. If A to B appears, right, and that's bad, then A to B is no longer possible, but B to A can now occur. And that's necessarily going to be good, according to the genetic entropy model. So that's a no. And then do populations experiencing mutations have uniform fitness? That's like the point is that they don't, because mutations are random, or at least probabilistic. So mutations lead to variations in fitness, and anytime you have variations in fitness, you could have selection. So the answer to all these questions is no. But if you go back and you read Sanford's book, his answer is yes to all of these questions. And not only is his answer yes, but the answer needs to be yes in order for genetic entropy to work. Genetic entropy doesn't work because the answer to all these questions is no. Again, we're not going to go into it any more detail than that because I have all this other stuff on genetic entropy. That's the short version of why genetic entropy is wrong, which standing just parrots unquestioned, without questioning it in this book, and doesn't address any of these shortcomings. So now let's talk about ENCODE. ENCODE is mentioned in a few places here. This is the project that sought out to map all the functional elements in the human genome and made headlines, many of them their own headlines, by saying that a large swath of the genome is functional. So down here on the left, let's see, we have strong evidence that the human genome is near fully functional. Near fully functional. And then here we have ENCODE yields, actually a full sentence. We have preliminary evidence for genome-wide functionality based on the ENCODE results. ENCODE has revealed that over 80% of our genome is active. You'll notice the switch in that sentence from functional to active. Those are not the same things, as I'll show you in a second from ENCODE's own numbers. ENCODE's own work shows that those are not the same thing. And then finally, over here on the right, we have a third different claim. In a nutshell, we now know that ENCODE has discovered that at least 80% of the non-coding DNA was transcribed into RNA, suggesting function. That's not what ENCODE found. The number varies from 62% in one study to 75% in another, not 80%. So these don't have sources that I can specifically go back, but I'm pretty sure I know where I'm able to kind of figure out some of these papers. So this is the most well-known ENCODE paper, the 2012 one. And this is what they find. 62% of the genome was transcribed. Overall, 80.4% of the genome has a biochemical activity. And in their kind of quick notes thing, they say these data enable us to assign biochemical functions for 80% of the genome. That's not accurate, as they actually say in the paper. They actually say that it's not 80%, but they're kind of like little tie-line summary says that. Not an abstract. They have like a little fact box that says this, but it's contradicted by the little paper it's attached to. So I'll figure on that one. They updated this in 2014 and basically kind of changed their way of presenting this to be a little more specific and basically distinguish between things like has a phenotype, in the red is conserved, is protein coding, and the different levels of biochemical activity. And by these new numbers, they were much more circumspect about how much genome is actually functional. Instead of reading you long quotes, I'm just going to show you the graph here. So what this graph shows is for different biochemical activities. So transcription, protein binding of DNA and then histone marks. So you're talking about modifying like chromatin packaging. These bars show different levels of activity, and the different colors represent different degrees of activity. So what you see for, if you just focus on this third bar here, that's all RNA transcripts. Well, this refers to the level of transcription, and anything from 0.5 to 5, depending on the cell type, is under 1 transcript per cell, which means it's not doing anything. And a lot of it's much less than that even. So it's clearly spurious transcription. So they have a total amount of transcription of about 75% of the genome is transcribed. That's a lot. But if you use the loosest criteria for like what is going to like actually have a function that they describe in the paper, it's only like 37, 38% of that is even potentially going to have a function, right? And you can do that for all this stuff. Come over here, it's like histone modification. This is I'm not going to go through the math of how I calculated this, but suffice it to say the white region is not statistically significant. It's activity, but it's like clearly spurious activity. It's not like a lot of activity. Well, what looks like 60%, that's a lot, but you got to lop off that top fraction. Well, now you're down to like in the mid 30s at most over there. So you can go and read papers from ENCODE and they end up just like saying completely the opposite of all the headlines about ENCODE. What they're actually saying is, yeah, the genome is very active, but in terms of functionality, there's a lot of it is not actually functional. And they actually say in the paper, we expect a lot of it to be spurious transcription. We expect a lot of it to be weak, just DNA binding that's more or less random, right? So the ENCODE themselves refuse this idea. Okay. Here, let's look at transposon function. This is a quick one. We're almost done here, everybody. So right here, Standing writes, another amazing observation regarding jumping genes and retrotransposons is that many retrotransposons have the starting sequence for many different genes. These jumping genes can jump around the genome, turning on and off genes performing functional roles. In other words, the transposons are active and they are functional in that, like in your body right now, and specifically it's in your brain, this goes on, where he's saying that when these transposable elements, these jumping genes mobilize, when they jump around your genome, that's part of the day in, day out normal functioning of the human genome, okay? Well, here's the paper that Standing posted to support this. Again, this is not in the book, he posted it somewhere else, and I tracked this down. So this is the line that he says supports his thing. Thus, somatic, and this is in the abstract, by the way, thus somatic genome mosaicism is driven by retrotransition, may reshape the genetic circuitry that underpins normal and abnormal neurobiological processes, okay? So he takes that and says, boom, these transposons are functional. Well, let's read the rest of that paper. We're not going to read the whole thing. I'm just going to point out the important paragraph. This is the important paragraph, okay? By contrast with germline activity, somatic insertions disproportionately impacted protein coding loci. Germline insertions are rarely found in regions where they generate a deleterious phenotype because such mutations are strongly selected against during evolution. In other words, for that sentence, what they're saying is the ones that you inherit, the transposons that you inherit, those are rarely in a region where they do anything, because when they do things, they tend to cause problems. So the ones that persist, they tend to not do anything. Somatic events, on the other hand, meaning when they mobilize, during development or something, on the other hand, are present for one generation and may affect protein coding loci at specific environmental context. The on the other hand is what matters there. Germline insertions don't cause any problems. That's why they're inherited. Somatic events, on the other hand, are present for one generation, meaning they cause problems and are selected against. That's why they only persist for one generation. So this paper that Standing quotes as evidence that jumping genes are just active as part of the normal regulation that goes on, in the paper that he writes for that, not in the book, because I can't figure out what the citation is in the book, in general, it says the opposite of what he claims. It says that typically they cause problems, okay? So like, it's just, again, it's an example of not understanding, either not reading or not understanding what is written, and then presenting it as the opposite of what it actually is. Okay, the last thing I'll talk about here is orphan genes. And I'm not going to quote anything from the book. There's a couple places where we make orphan genes. But the thing I want to point out here is that, one, we know where orphan genes come from. We figured this one out. 20 years ago, this was a bit of a mystery. we've now figured this one out. The answer is that you have long non-coding RNAs that acquire the ability to be translated. Basically, you get a start codon. And there's lots of evidence that essentially random sequences can be biologically functional and relevant and have lots of cool activities. And they're also very well tolerated when these random things are expressed in cells. They don't often cause problems. So this leads to a really nice kind of avenue for new genes, de novo genes. Well, what we can do is we can look genetically and say, okay, we have a new gene. Do we have a related species, any non-coding RNAs that match to that? Oh, we do. That explains that orphan gene. Okay, that's what's going on there. Now, the contradiction here is that the creation model, and Stanning says this in the book, the creation model predicts nested hierarchies and it also predicts orphan genes. Well, which is it? It can't predict both, right? Either nested hierarchies, what we expect from special creation, or differences within each group due to their independent creation or what we expect. Is it, which is it? One or the other, right? So there's the contradiction that Stanning doesn't explore at all. He just presents both sides of it without seemingly realizing that there's a contradiction there. And here's another problem. Orphan genes are typically limited to species. That's why they're orphans. They're in one species, but then they're not in the related species. Well, young Earth creationists typically accept common ancestry within families. So you can't, as a young Earth creationist, turn around and say orphan genes refute common ancestry, but then also accept common ancestry within families. If you take that position, then you're locking yourself into a situation where species are the created kinds, not families. And then, of course, that leads to all sorts of problems with, like, space on the ark, for example. So there's a bunch of contradictions that creationists are faced with when they deal with orphan genes. None of those are, it doesn't seem that Stanning is aware of those contradictions in the way that he presents orphan genes in the couple of places in this book where they are mentioned, okay? I think that's every topic. There's a few that I left out, but I think, yes, that is every topic that I wanted to cover. My goodness, that took almost an hour. I'm sorry, everybody. I did not think it was going to take that long. But that's at least a rundown of the science in this book. And basically the arguments that Stanning makes, I don't want to say they're his arguments because they're not. They're lifted from professional creationists. But that rundown of the argument that Stanning makes is why they are wrong. Basically, what this book contains is a bunch of creation science from professionals, Sanford, Jeanson, Carter, other people, plus a bunch of quotes lifted from biologists they say things like, oh, I'm skeptical of the ability of evolutionary processes to do X or whatever, something. And they'll say, oh, this actual biologist says that evolution is false. But if you ask that person, like they don't think that people do a good job of explaining how different processes work in very specific contexts. One particularly egregious example of this is I'm forgetting the biologist's name off the top of my head, and I apologize, everyone. Someone in the chat, tell me, who was it that came up with the theory of endosymbiosis for like mitochondria and chloroplasts? Lynn. What's her name? Here we go. Lynn Margulis, okay? He's got a quote in here from Lynn Margulis saying, evolutionary process, whatever. Although random mutations influence the course of evolution, the influence is mainly by loss, alteration, refinement. Never, however, did one mutation make a wing, a fruit, but blah, 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 blah. Basically saying, thank you, that she says that evolution can't work. She came up with the idea of endosymbiosis. That's one of the most important evolutionary processes in the history of the development of life. That's how we got mitochondria and chloroplasts. To present her as somebody, she does have a lot of weird ideas, but she is not a creationist. And to present her as supporting creationism by lifting a quote out of context, and I'm not often one to accuse people of quote mining because I think that accusation is thrown around too loosely, but that is a quote mine. That is taking a quote out of context to make the person saying the quote appear that they mean the opposite thing of what they actually believe. Okay, so that is there's several examples of that. By the way, Rational Mind, I don't know if you were here when I used one of your figures from your delightful series. So thank the one with the mutation rates and substitution rates as they decrease over time. So thank you for that one. That figure is the clearest I've ever seen of that. I agree. Quote mining is lying. Quote mining is fundamentally dishonest, and I will say there are parts of this book, like that instance, that are fundamentally dishonest. So that's what we get. We have creation scientists that stuff is taken kind of through SFT's filter. The quote mines are taken through this filter. There's a fundamental lack of the understanding of the basics of biological processes, like whether mutational meltdown is the same as genetic entropy or the opposite thing. So you end up with a bunch of arguments that are not just incorrect if you parse the arguments carefully. I've gone through this, and I've like, what I've presented here is like, this is what standing for truth means as far as I can tell. But in terms of the way he makes the arguments, it's not just he's making bad or incorrect arguments. They are often incoherent. They are sometimes self-contradictory. It's kind of a mess, honestly. And it leaves me wondering what the goal of this work is, because as a work of persuasion, I don't think it is effective. I think some of you here know Corporal Anon, our mutual friend. I think he said it best, that this is the point here, is this is a dopamine hit for younger creationists. This is not meant to be a work of persuasion. This is not meant to present a serious argument. This is to make creationists feel good and be told that no, trust me, there is science that supports your position. Now, none of that science, as we've gone through here, is actually correct, but that is, I think, the point of what's going on here. So that concludes my review of Universal or Separate Ancestry, the biblical model of origins made easy, the question of ancestry finally answered by standing for truth. That is the we'll say the more technical review of this book. It's not complete, but that's a rundown of the actual science in the book and why, like all creationist science, it is completely wrong. So goodness, it's already 9 o'clock. Let's anybody have any quick questions for like next 5 minutes ish, and then we'll call it good. Is everybody hanging in there? So there's still 20 people here. Wow, thank you all for listening to this for an hour. Goodness gracious. Let's see. Is anybody we got any? Okay. So I think we're gonna call it good. I think thank you for watching, everybody. I hope you enjoyed. I hope you found this useful and informative. And next time Standing and any of the Brain Trust brings up any of their stuff, you got the counter arguments. They're right here. Let's see. Oh yeah, I just totally ignored the human chromosome 2 thing. So the gene that they like to point out for human chromosome 2 is, there's a gene that transcription spans the fusion site. Human chromosome 2 is the only place in the human genome where that family of pseudogenes, it's the DDX11L family, I think. The only place in the genome where they're not located immediately next to a telomere is in human chromosome 2 at the fusion site. It's really easy to have transcription across that gap. That's not hard to accomplish, so that's silly. What would happen to the diversity of a hypermutating but inbreeding population? Well, if it's inbreeding, then it's decreasing, definitionally. The definition, like the way you look at a population and see if it's inbreeding, is that it has lots of homozygosity, more than you would expect based on the population size. That's how you indicate inbreeding. That's what happens. Now, hypermutation has increased the genetic diversity, which definitionally increases heterozygosity. So those are opposite things. Thank you, Dapper, for that question. David, hit it. What you got? And I'll figure like 10 after 9. We'll call this good. If you got any questions, we'll go until then. Don't want to make this one of those long streams like some people. Oh, that was your question. Okay, cool. Yes, and it is the case that the only place where we find head to hair telomere repates is the fusion site. Yep, it's plus the fact like put aside the fusion site. Let's just ignore the fusion site. Okay, let's pretend the fusion site is non-existent. Let's just cut that whole thing out. You take the 2 chromosomes from the chimp and you line them up, and you get the human chromosome. Okay, it's all there. Like, it's just boom, boom. Like it's there it is. So like you just have to ignore the whole rest of that thing. Neanderthals had a lot of genetic diversity rather than little. Could we still say they were outside Homo sapiens? Yes, because diversity and divergence are two separate things. Thank you for asking, Dapper. So diversity refers to the number of differences or the degree of differences within a lineage. Divergence refers to the difference between two different lineages. Neanderthals are divergent from Homo sapiens. They are two separate lineages. That's different from the diversity within either of those two lineages. So even if you were to invoke hypermutation in the Neanderthal lineage, that doesn't solve the divergence problem. There are SNPs, single nucleotide polymorphisms, that are found in humans, but not Neanderthals. There are those that are found in Neanderthals, not in humans. They have different sets of genetic diversity. All right, let's see. Are herbs functional? Some of them are. A very small number, 8% of the genome is herbs, 8 to 9%. A very small handful are functional, and it's usually just promoters that are useful, not the GAG, POL, and M genes that make up the intact herb. It's the promoters that we do use in some situations. Humans do use herb promoters occasionally, but those make up a very small fraction. So if you want to claim that herbs are functional, fine, you'll find biologists agreeing with you, but that the fraction of the genome that's going to be covered by that, I mean, out of the 8% of the genome that's herbs, most of those are degenerated. The ones that are not, that are intact enough to be useful, let's be generous and say it's 1% out of that 8%, so 1/8 of them. And then within that, you're only looking at the promoter sequence, not the whole thing. So it's a teensy tiny fraction that would actually be useful. How do they become functional? Co-option. They land somewhere in the genome, and then we kind of deal with it, basically. Thank you. Let's see. Let's see. Sorry, the bunch just went fast there. Herbs are functional and not even evidence. He's saying that they're created. He's saying the herbs are created in there. And yes, the fusion site is where we'd expect it. Yeah, it all just goes together very nicely. Let's see here. What else do we have? Evolutionary botany, because I'm not a plant biologist. I apologize. How do we know herbs originate from viruses? Because they have all the virus things. They've got the GAG, POL, and M genes. The promoters look like viral promoters, not human promoters. They have the tandem repeats on the ends, the terminal repeats. Like they're viruses. It's not hard. Say genetic entropy is essentially a useless argument. Yeah, it's pointless. Genetic entropy is not valid at all. There's nothing correct about it. Like, it's that simple. There's nothing correct about genetic entropy. Creationists should not use that argument. No, plants are awesome. I didn't study plants, but plants are really cool. I think we're good, everybody. We're going to call it a night. All right, I'm going to call it a night. Thank you, everybody. I appreciate you all being here, watching, asking questions. I don't know about turtles and phylogenetics. Sorry, I'm really bad on like vertebrate stuff, so I apologize. If creationists can't use false arguments, what arguments would they have left? Then they wouldn't have any because they're wrong. So there you go. Virology is also my favorite, or one of my favorites. All right, thank you, everybody. Have a great evening. I appreciate it. Appreciate you all being here, watching, and asking me questions. Have a great night. Bye.